Welcome, everyone, and thank you, everybody, for joining us today for today's webinar. I think we have like a minute more to begin, but while we're waiting for the others to join in, just a couple of topics of housekeeping. My name is Rashmi. I will be the moderator from Protein Fluidics, and I will be answering your questions live as they come in. Along with me is Jackie Boland, who's a technical and sales specialist from Greiner Bio 1, who will also be moderating the questions. So feel free to ask your questions in the chat box on the right. Your mics and your cameras are turned off, so you can ask questions only via chat, but we will be asking your questions live as well. And without further ado, it's my pleasure to introduce our first speaker, Dr. Glauco Souza. He's the Director of Global Business Development and Innovation in 3D Cell Culture at Greiner Bio 1 and former President and co-founder of Nano 3D Biosciences, Inc. He is one of the co-inventors and inventor of 11 patents related to magnetic 3D cell culture, including magnetic 3D bioprinting and levitation technologies, and has authored numerous publications in this field. Dr. Souza is also an adjunct assistant professor at University of Texas Health Science Center at Houston. Prior to founding N3D, Dr. Souza was an Odessi Scholar at UT MD Anderson Cancer Center. He has received his bachelor's, master's, and PhD in physical chemistry from George Washington University in DC. Without further ado, and thank you, Dr. Souza, for this talk right now.
Thank you very much, Hashimi. It's great to share some of our results, and of course, this is a great opportunity to have a joint webinar with Protein Fluidics, Evan and Rashimi. I'll go right into it. And of course, first thing, I will start with acknowledging the people that helped us, the organizations that funded us. Most of the work I'm going to share with you was funded by NIH, by NSF, by CASES, Center for Advancement of Science in Space. And we have several collaborators that really helped us to get where we are. And this goes also to, I want to touch on the origin of technology, where everything started. So this is from my background that Hashimi mentioned. The technology came from a collaboration between Rice and MD Anderson. I was at MD Anderson, and I was at Odyssey Scholar MD Anderson, where we came up with the technology. We started magnetically levitating, bioprinting cells. Then we started a company called Nano 3D Biosciences, and almost 3 years ago, Greiner acquired the technology, and which is why I'm with Greiner now. So I love this quote because, of course, from Albert Einstein, but everything should be made as simple as possible, but no simpler. So the reason I like this quote is because that's the way I visualize magnetic 3D cell culture within the field of 3D cell culture. You know, we can simplify. So if you go from 2D to 3D, you're going to have to add complexity. But we try to make it as simple as possible by introducing the magnetic field for us to manipulate and facilitate cell culture, 3D cell culture. And of course, I'm going to be very brief because I'm sure I'm preaching to the choir, but we always have to answer why 3D cell culture, right? We've cultured cells for a long time. We put cells on a petri dish, they stick to the bottom, they grow as a monolayer. We know how to do this. But one aspect that 3D cell culture is really impacting, and I'll show some of these results as well, is on drug discovery. We know drug discovery is expensive. It's a long process, and part of the reason that it's expensive and it's a long process is expensive is because most drugs, when they get to humans, they fail due to poor efficacy or toxicity. So, and part of the reason for that is because the initial process of drug development, cells are cultured in monolayer, and those are not very good representation of in vivo biology. You go to animals don't have human physiology, and no surprise, drugs fail when you get to humans. So with 3D cell culture, we're trying to bridge the gap in using human cells to get better results and more predictive and make the drug development cheaper and faster. But if you really go into, some of the practical aspects of 3D cell culture, one thing that you realize when you do 3D cell culture, or when you go from monolayer culture, 2D cell culture to 3D cell culture, is how important it is cells attached to plastic. And that's one thing that you lose. When you go from monolayer culture to 3D cell culture, you lose the cell attachment, and all of a sudden, simple tasks like changing media, which is very simple, is not simple anymore because cells are no longer attached to plastic. Immunohistochemistry, immunofluorescence staining, and you will see also on results from Evan. When you're doing 3D cell culture, and you are not able to attach cells to plastic, that becomes very difficult. So we're using the magnetic field as a surrogate to cells attached to plastic. So that's how we're going to be able to make manipulation in 3D cell culture as easy as in 2D. And of course, first thing, cells are non-magnetic. First thing you have to do when you're doing magnetic 3D cell culture is magnetize the cell. So we have the NanoShuttle, the nanoparticle assembly that just sticks to the cells electrostatically. And then we use the same protocol as you do with 2D cell culture. You detach, you count your cells, and you can place the magnetic field on top, and the cells will levitate and assemble in 3D, or you put the magnetic field on the bottom, and you can really fine-tune and bring the cells together and localize the cells within the well. So this is just a quick view of different results we have. And one thing that with magnetic 3D cell culture, you can magnetize the spheroids and use the magnetic field to bring them together, which is what we're doing here. Here we magnetize a GFP label glioblastoma, and then we had M-chair label astrocytes. We cultured them separately, and then we magnetically brought together, and you can see the glioblastoma invade the astrocytes. On the bottom right here, you have a co-culture of endothelial cells and pre-adipocytes, where we differentiated pre-adipocytes. The red is prolipin, which is a marker for adipocytes. The green is GFP label endothelial cells, and we control the rate by controlling the ratio of the two cell types. We're actually able to see the sprouting and the vascularization of the endothelial cells. And this was the cover of Tissue Engineering a few years ago. The bottom line is, by being able to magnetize the cells and use magnetic field, you can do a lot of things that are easy to do in monolayer culture, but it's not as trivial to do when you're dealing with 3D cell culture. And of course, and here is the cover of Tissue Engineer on the right that I mentioned. And I like to show this slide for a few reasons. Magnetic 3D cell culture is a relatively new technology, so the best way to validate a technology is by publishing. And we've published very high-impact journals, very good journals, and worked really hard, especially as a small company as we were with Nano 3D Bioscience. We're really focused on publishing in good journals. But the other reason also that I like to show this slide is because we use the nanoparticles. And it's a fair question. What do the nanoparticles do to the cells? And we've shown over and over. The reviewers always have asked that the nanoparticles basically don't do anything. It's bioinert. And we've shown over and over that it's the nanoparticles have no effect on the cell biology. And we have a 5-page white paper that we compile the results: viability, proliferation, protein expression, stem cell differentiation, inflammatory stress, oxidative stress, gene expression with and without the NanoShuttle. And we show that there's no difference. So the technique is very simple. It's basically a magnet on the bottom of the well. As you can see, you have a drive, you have your well. And once you magnetize the cells and put the magnetized cells in the well, they come towards the magnet. And you do not need to leave the cells on the magnet to then assemble. You just have to promote cell-cell interaction as few minutes as 15 minutes, and remove the magnet, and cell-cell interaction takes over and form the 3D assembly. So I want to show you a quick movie that shows the exactly workflow of this tool. Let me see if I can. So you have, it's a kit. So you have your NanoShuttle, which is nanoparticle assembly. You basically just pipette the nanoparticle to the cells. The cells are attached. This NanoShuttle will just settle on the particles and will label them that way. It's a very simple. Then you're going to wash your cells with PBS, remove trypsinize, and count your cells, all the same. And then you're going to put your micro-L plate on the magnet drive, dispense your cells, and as soon as the cells see the magnetic field, because they're magnetized, they will come towards the magnet. And 15 minutes on the magnet, and you'll see the cells are all in the middle. And this is a flat well, so cell-cell interaction will promote the formation of the spheroid. Now, of course, if you want to add a drug, you want to replenish your media, you put it on the larger magnet so your spheroids are tight, you remove the media, you add your drug, and that's it. So that's basically the workflow. And we like to say that we are, here you see 9638415536 format, and we like to call ourselves a 3D and a 2D workflow. So, and I want to show you some results of some using our high throughput system, which was a collaboration between Scripps Research Institute and Cold Spring Harbor, where we got an NCI grant to validate our high throughput screening system. And this is a 1536 drive. As you can see, very dense, lots of little magnets. Every magnet sits in the middle of the well. And as you can see here, this is colon cancer, HCT, colon cancer and lung cancer cells. On the middle here, the cells are just in the well without the magnet, they spread out on the well. You put on the drive on the magnets for 15 minutes, and you can see the cells come together and they start forming the 3D assembly. Just a picture, and it's hard to see here, but you have blown up. Image here is just to show that you have 1,536 wells, and you have a very uniform spheroids, uniform in size spheroids made on each well. Now, this is one aspect of 3D cell culture that we try to address. One challenge of 3D cell culture that we try to address using the magnetic field. So when you're doing high throughput screening, and you want to use a flat surface, right? Because flat surface, you're going to get the best images. And your spheroids are in the well, as you can see here. Every dot here in the middle of the well are spheroids. Thermal motion usually makes the cells to flow to the side, and they stick to the wall. And when they get to the wall, it's very hard for you to collect that data. So basically, if you wanted to do imaging as your readout, as your endpoint, once the spheroids are on the well, it's a data point lost. So what you can do is put the plate back on the magnet, and you can see 30 seconds on the magnet, and you can recenter all this position and recenter all the spheroids where you want with the magnet. So this is a very useful tool to position the spheroids and expedite the imaging and speed up your results. So again, we're talking 3D cell culture, and of course we have to show the image of spheroids. And here is an image of magnetized spheroids with different cell types. You have HT29 and pink one. Those are cell lines. HT29 ovarian cancer cell line, pink one pancreatic cancer cell line. And as you can see, you bring the cells together. You see 4 hours here, and over time they make beautiful spheroids. So now, just to get into a little bit on this study, the work we did with Cold Spring Harbor, I mentioned that was a grant from NCI to validate our high throughput screening. But we also, on this work, we use human patient cells. And in AGM1 and H21 are two different patients. In AGM1, we call here is the pancreatic cancer cell, and AGM1 CAF is the pair, is the cancer-associated fibroblasts from the same tumor. So Cold Spring Harbor took the tumor from the patient, separated the CAFs from the cancer cells, and they actually expanded the two cell types. And then we magnetized the cells. And as you can see, the CAFs, the cancer-associated fibroblasts, you bring the cells together, they make beautiful spheroids after 72 hours, actually after 24 hours. And then when we look at the cancer cells, the AGM1 and HT1, you magnetize the cells, you bring them together, but they do not form one single cohesive spheroid. They actually form organoids. And this is recharacterized. We published this in SLS Discover a few years ago. And you can see the organoids have the cystic type of morphology shape, and they have the polarized cells. So even though they don't form a cohesive spheroid, when you bring the cells together, they form these organoids. By magnetizing the cells, you can still use the magnetic field to center them, to manipulate, to change media. So there's clear benefit of using the magnetized spheroids, even when we're generating organoids. Now, to get into the results of this work, which was pretty cool, what we learned from this study. Part of this grant that we had was to validate our 3D cell culture, high throughput 3D cell culture system, and also compare 2D versus 3D. And part of our hypothesis is that 3D cell culture would give us less false positives than monolayer culture. And the reason for that is because with the 3D shape of the culture, diffusibility of drugs, and all the aspects of the cell biology that is promoted by cell-cell interaction, cell-ECM interaction, also play a role on the drug effectiveness. So what we did to prove the hypothesis, we took 3,300 compound library, FDA library, which we did not have cancer-prescribed drugs, which means if it was a positive, if the drug actually killed the cancer cells, we took that as a false positive. So, and here are the results. So in the middle, we have 2D, a number of active compounds in 2D. On the right, you have a number of active compounds in 3D. And here are your patients' pancreatic cancer cells and pancreatic cancer-associated fibroblasts. And as you can see, for the total count in 3D of these 3,300 compounds, we always had less active compounds in 3D than 2D. So in some ways, we are validating that hypothesis that you do get less false positives when you're working with the 3D cell culture. So another way of looking at this data is we plotted 2D inhibition on percentage inhibition on the x-axis and drug inhibition in 2D on the x-axis and drug inhibition 3D on the y-axis. And so every dot you see here is a compound. So if the compound is on the white quadrant, that means that the drug didn't really do anything to the cells. So, which most of the compounds of these 3,000 compounds, most of this, as expected, were in the white quadrant. They didn't kill the cells. So if you go to the pink quadrant, are the drugs that are killing the cells or effective in 2D only. The green is the overlap, is where you have the compounds that were active in 2D and 3D. But what was really cool from these results is that the most active, only a few drugs were active in 3D, and the most active one was disulfiram. And disulfiram was actually in phase 1 clinical trial for pancreatic cancer, which means that they had already determined that this cell was active in vivo, and it did not do anything to the cells in 2D. So not only we reduced the number of false positives, but actually we picked up the needle in the haystack, the jam from this screening. And disulfiram is acetaldehyde dehydrogenase, and it's a drug used for drug addiction and alcoholism. Apparently, it's a pretty nasty drug. So in this, we're going to publish very soon. These results are not published. We did 150,000 compound screening. For those of you interested in compound screening, the number here to keep in mind is the Z prime. The Z prime is 0.72. which is, as we know, Z prime above 0.5 means it's excellent. The Z prime is an excellent assay. And one important thing too, we use here cell titer glow. So the Z prime is based on cell titer glow, and of the 150,000 compounds, we picked the 735 leads, which is about 0.5% of the compounds were hit. And this we're going to publish really soon. I'm not going to get very much into this. I also want to touch on what is the direction of the field that the field is going, which is precision medicine, right? Personalized medicine. And here's some results we did with UT Medical School in Houston. And we took a needle biopsy of metastatic prostate cancer metastatic bone mat. We're able to take the bone mat, break it apart, magnetize the cells, bring them back together, and within 96 hours, we got a very nice dose response curve to doxorubicin, which it is the drug physicians recommend for EpCAM positive prostate cancer. We also put the cells back on the magnet, and we're able to stain for EpCAM. And Evan on the next presentation is going to show some pretty nice results taking this similar approach as far as doing immunohistochemistry. Last topic I want to touch on, it's a new tool that is coming soon, and Evan will also use this approach, is using the magnetic field to actually transfer and move spheroids. And we call the multi-mag pen. So the multi-mag pen is basically a magnet array that we have a 24 and 96 well. So there are magnets that fit right into 96 or 24 micro well. So you have your, what we call the multi-mag pen drive. You have a sterile sleeve, which goes between the magnet and the culture. And you have your cell repellent plate, which will have your magnetized spheroid in there. So when you assemble that like that, you can actually pick up the spheroids very easily. As you can see the spheroid, this is, you have your plastic sleeve, your magnet, that is your plate with spheroids, and you can pick up our spheroids, and you can easily transfer to a clean well. So this is a very cool tool, and we have also the single mag pen tool to transfer spheroids from well to well. And we call bioprinting. So I always like to show this slide because, again, people say, oh, you're just bringing the cells together. What do you call bioprinting? Because we can use the magnetic field to bioprint. And here we just wrote in for NanoShuttle using GFP labeled glioblastoma, and we can do much more complex structures by just playing with the magnetic field. But last, I'm always very proud and this work we did in the space station. This was work funded by CASES, Center for Advanced Science in Space. And this is Peg Whitson working with our system in space. And people usually ask me, why are you using a 3D cell culture system in space? There's no gravity. Cells grow in 3D in space. But it's very hard to manipulate cells in space. By this, we send the magnetized cells to space, and we cryopreserve, they're already magnetized, and then we just were able to manipulate using the magnetic field and make the 3D cell culture more easily. And that's all I wanted to share with you for now, and I'll pass the presentation now to Evan.
Thank you so much, Dr. Souza. So I have a quick question. So have you ever tried using the NanoShuttle for studying organoid to organoid interactions? Yes,
and there is some results that I did not show that we did with scripts. We have, and we also have done spheroid to spheroid interaction, which is I showed very easy. There was we published some of this data in Nature Nanotech and Neoplasia early on, which we co-culture glioblastoma and astrocytes. And okay. I'll be glad to share some of those results.
Thank you so much. And thank you. And while waiting for more questions for you, I want to go on to introduce our next speaker, Dr. Evan Cromwell. Dr. Cromwell is President and CEO of Protein Fluidics. He has 30 years of senior executive and technical experience and has authored over 40 publications in the fields of biology, chemistry, and optical instrumentation, and holds 16 patents. Prior to founding Protein Fluidics, he served as a director of research at Molecular Devices and was the leader of cross-op scientific team for Danaher Life Sciences. He was also the founder and president of Blue Shift Biotechnologies, which was acquired by Molecular Devices in 2008. So Evan holds a bachelor's degree in chemistry from Caltech and earned his PhD in chemistry from UC Berkeley. So without further ado, Evan Cromwell.
Well, thank you for the great introduction, Rashmi, and thank you, Glauco, for the presentation. It's always interesting to learn about the latest that you're doing with your NanoShuttle and magnetically bioprinted organoids, spheroids, tumoroids, whatever kind of oids you want to call them. Let me talk first about who's been helping us here. We're a small company, a young company like Glauco was 10 years ago, and we have a group of protein fluidics, including Rashmi, Anthony Tai, and Katya, who's our new application scientist. All the people at Greiner, a lot of help with molecular devices, especially Oksana Sarenko and Matthew Hammer. A lot of the images I'm going to show you are from them. Tulane University, they're the ones with the patient-derived tumoroids and all the cell lines and the great data I'll show from those cell lines, and then the groups at Promega, which are helping us on the assay applications. So today I want to talk a little bit just about use of 3D cell models. I think Glauco gave a really good overview of why 3D is superior to 2D, and I think the whole industry is sort of moving this way, although kind of slow at times. But then, how do we use our Puma system? How does that work with that? And then specifically talk about applications with magnetized cells. May the force be with you for all you Star Wars fans out there. So talk about Puma System 3D Mag, and then two applications, one with cancer spheroids and then one with patient-derived tumor. So when I think of 3D cell models, my background being in the drug discovery industry and the high throughput screening, they are using 3D cell models and spheroids in high throughput screening. Glauco showed from Scripps a 1536 well plate, really amazing screen and engineering feat to do that. But you're really doing high number of samples analyzed, but low number of data points. Whereas if you look at some of the really amazing organoid chip, they're doing one or two organoids, but collecting a huge number of data points, a very complex model. And so you're really going from a simple model to a complex model, large number of samples to low number of samples. But you think there's a sweet spot in the middle where you want to do medium number of samples and you want to collect a number of data points from that. We call it multifunctional profiling. And so that's where we've really designed our Puma system to sit in that area. And when you think lab on the chip technologies, again, there's a lot of very complicated devices which you do very sophisticated measurements, but you have a lot of connections you have to do. They do have the smaller volumes and reagents, but devices themselves can be very expensive to manufacture and make. And then these tend to be single purpose, so you're really spending a lot of time to develop all the intricacies of your microfluidic device to do one thing. And if you want to do that thing, that's great. But if you want to do general things, have a more open platform, then it's more difficult to use those systems. So that's what we're really focused on with the Puma system, with the open lab on the chip for doing organoid assays. So you can maintain physiological conditions, you can grow different types of organoids in them. Now, in the system we have right now, the data I'll present to you, the organoids, tumoroids, spheroids are formed outside the system, you transfer them in, and that's actually where the NanoShuttle becomes very useful to help to do that transfer. But you can range from small 100 microns up to millimeter type size organoids, and we automate the assay steps for you. So that really takes away a lot of the complexity, and running the assays allows you to get better reproducibility, lower your CVs for your assays when you're running them with these really precious organoid samples. So the workflow we've developed is Flowchips, I'll show in the next slide, are loaded with your organoids and reagents. And then those are placed into the Puma system, which can reside in your incubator. So you can run all these assays at 37 degrees with 5% CO2. The protocols for running your assays are preloaded on the system. So it's microprocessor controlled with a pneumatic manifold system to drive fluid from one point to another, hence the acronym PUMA, or pneumatic microfluidic assay. And then when you're done running those assays, your samples are ready for analysis. So you can image them, high throughput, high content screening, you can read luminescence, you can collect supernatant for downstream analysis. And so it's a wealth of information you can then get from your organoid samples after running the assay protocols. Now the Puma system itself, the Flowchips, are based on a design which really lends itself to your existing assay and laboratory automation. So the wells are on the 384 well footprint, spaced 4.5 millimeters apart, and it looks a little like a 384 well plate, except what it is a series of 4 chips, and each chip has 8 tests. And each test is 1 lane, and so it's a series of wells that are connected by microfluidic channels. The center well here with the large aperture is where your sample is located, and then you can use the other wells as reservoirs for media exchange, compound addition. You can use it to sample supernatants. You can do lysing if you want. So there's a wide variety of standard assay steps that you can do using these wells. The wells themselves are small volume, so 20 microliters. And then the organoids or spheroids are located at the bottom in what we call a protected sample chamber. And we just recently published in SLAS Technology, EPUBs available online. So this protected sample chamber is one of our key proprietary technologies. We have several patents issued on this and several patents pending, both US and international. So the organoid is dispensed into the well, and then it's located down at the bottom in this chamber. And what that allows you to do is to transfer fluid in and out without disturbing your organoid or spheroid. You can exchange up to 95% of the volume. Volume down here is about 1 microliter, and total volume is 20 microliters. The aperture here is roughly 1.2 millimeters. It's a clear bottom, so it's a thin COC bottom plate. In fact, this whole chip is just injection molded COC, and so again, very, very cost effective. But because it's a thin COC bottom, compatible with very high resolution confocal imaging, you can get amazing images and amazing data from your organoids in our Flowchip. So I mentioned it's microprocessor controlled, so everything is self-contained. Protocols are preloaded, so if you want to incubate your spheroid 24 or 40 hours, etc., you can just select that protocol and run it. It also allows you to do your protocol step by step. So let's say you wanted to incubate for 24 hours, take out the plate, image it, put it back in, and run another 24 hours, maybe with a different compound or different additions. That all can be done, by creating individual protocols for those assay steps. So then, very flexible, very easy to use system. Again, thinking about where the sweet spot for our system is, it's really automating specific assay protocol steps. We don't form the organoids in our chips, not yet, but we do automate things like doing compound addition, doing time course sampling in situ. So you don't have to take the sample out, especially if you wanted to run shorter time sampling, 30-minute, 60-minute sampling. You can do sequential drug addition. You can do in situ cell lysing. If you're interested in metabolism, you wanted to capture metabolism with the organoids at 37 degrees Celsius. You can just shoot in the cell lysing solution and freeze that organoid and the metabolites in place. And then also immunofluorescence staining. And that's really difficult when you're trying to do this manually because there's so many steps involved, from adding your primary antibody to doing multiple washes, your secondary antibody, etc. So we can automate all that because the organoid spheroids are located in that protected chamber. We can do that without loss of sample. And then in terms of the different types of organoids we've worked with, I'll show you examples here of cancer spheroids and patient-derived tumoroids, but we've done work with neuroscience, mnemonics, microbrains. We've done work with different iPS-derived organoids, lung organoids, etc. So a wide variety of sample types possible. The 2 types I'll talk to you about, the 2 applications, are one with single-cell spheroids and then with primary tumoroids. And some of the other data we have, we'll probably present in another webinar a little later in the year, is really working with intestinal organoids and organoids that have been grown in matrigel and then transferred into our Flowchips. But I won't be talking about those too. So cancer spheroid profiling. We like using the term multifunctional profiling because it really is the strength of our system, where you can use your organoids, your pressure samples, and then do multiple types of analyses for them. So if you want to incubate with a compound, you want to look at the viability, they call it the kill curves, if you will. Doing live data analysis, it's very straightforward. You can look at phenotypic changes of the organoids. You can also pull those supernatant samples and look at secreted factors from those samples and do this at different time points during your assay protocol. Now, where we have really utilized this magnetic particles and the NanoShuttle is in helping to transfer and position the spheroids into our wells and hold them in the center. Glauco showed how you can use magnets to pull them into the center to improve your imaging. Same is true in our system. If you just let these little organoids sit around, they're going to roll to the edges, and if you want to do really nice high resolution confocal imaging, it's a little harder to do that with them shoved against the side. But with the NanoShuttle material, you can do the magnetic bioprinting of your spheroids, now magnetized. When you load them into the plate, we have a little magnet at the bottom here. We have a specialized loading tray, our 3D mag system, and they'll pull them down and center them in the bottom. And you can see here an image of it's actually a microbrain that we had magnetized. See it nicely centered in the well. Point out the little points of light here. These are the side ports where you can transfer the fluid in and out of the well, 7 of those side ports there. Glauco also mentioned the 3D mag pen, and you can imagine how that would work here, where you can then load and attach the organoid or spheroid to the mag pen, lift it up, you can pull it over, lower it down into the bottom, and then by pulling up the magnet, spheroid gets released and held into the bottom. And so this again allows you to do that transfer, and you can even imagine doing this in automated fashion where you can pick up all 32 spheroids at one time, transfer them over, drop them off, and now you're ready to go. So it really takes a lot of the pain of trying to pick these up with a pipette by aspiration, and then dispense them into the Flowchip. Really an elegant way to use the magnetic force to automate processes which can be tricky if you're doing them manually. Okay, so these are HeLa spheroids, about 4,000 cells. These were magnetically bioprinted with the NanoShuttle material, and then they were placed into our Puma Flowchips. We did a number of different types of assays with these, but in this case, these are just stained with a cocktail of Hirsch mitochondrial stain and a NucView apoptotic stain. And this is just showing you the type of images you can get not only from the clarity because of the Puma Flowchip, but also the lack of any interference or any artifacts from the NanoShuttle. So you get very nice resolution here. You can see cell cycle mitosis, etc. NucView, you can count number of cells in apoptosis. So it really is no interference from the NanoShuttle. You get the benefits of being able to form the spheroid, the benefits of being able to transfer them, no interference with your biology. And this is from the SDICE technology article that we've just published, and just showing 2 different types of spheroids, HeLa and HepG2, where you can look for cell viability, and you can look for using image express and MetaExpress analysis, do cell counting for total, live, dead, apoptotic cells. And then this really shows the multifunctional profiling, and now you can combine that looking at the response of different spheroids to treatments, as well as pulling the supernatant, in this case measured VEGF. You can also look at cytokines and any other secreted factors. But an important point is you're looking at single spheroid secretions. So now you can imagine doing single spheroid type analysis or single organoid type analysis, where not only you look for response of those compounds phenotypically to treatments, looking at the cell response to those treatments, but also secretion metabolite response to those treatments on an organoid by organoid basis. And so for organoids, then I really think tumoroids is the term we use for these patient-derived cell lines. And this is work that we're doing with the Tulane group, Matthew Berger's group, and specifically Marguerite Matosin. This particular line, I think they have like 10 different cell lines that they've taken from triple-negative breast cancer patients. Particular line 4IC was a patient that exhibited resistance to paclitaxel or Taxol therapy. So Matt's group does both the PDX, as patient-derived xenograft models, and the patient-derived tumoroids. In this case, these were the 4IC cells which were cultured up and then formed using ultra-low attachment plates for 72 hours. And then we coated them with NanoShuttle after that. So in addition to doing sort of the single tumoroid formation, we found using these microwell array plates, in this case, the 24-well agro-well plate from Stem Cell Technologies, really provides an advantage if you want to create large number of these and large number of fairly uniform tumoroids. So in this case, the tumoroids were grown in one of these microwell plates, 48-hour incubation, and at the end of you have 1,200 tumoroids coming from this single well. You can then add the NanoShuttle on top of it. I joke a little pixie dust on top of that, and then just incubate that for a couple hours. You can then harvest all those, so they come out of the microwells very easily. You can then put them through a strainer to rinse away any other media or any excess NanoShuttle, and then transfer them to a little tube. Once they're in the tube, you can then pipette them up and dispense them down into your plate. Again, using the magnetic properties of the NanoShuttle to help transfer and center the tumoroids in the plate. This is just shows examples of 3 sort of representative wells. Each one of these is a tumoroid. They're roughly 150 to 200 microns in size. They've been transferred in. You can see how they're nicely centered in the well, healthy, nice translucent type appearance to them, and kept away from the sides. So again, this is the strength of the NanoShuttle, allows you to magnetize it and do manipulation, do loading, do centering of these tumoroids to really improve your assay, provide you superior results. Okay, so the type of work we've done with this, so some of the examples here are, again, this multifunctional profiling. We're doing viability assessment, looking at metabolite analysis, and then doing immunofluorescence staining of these tumoroids. So from the viability, we used the Puma system and the Flowchips to do a compound incubation. This is doing overnight 24-hour or 48-hour incubation, compound in there. And after that, you remove the compound and replace that with the stain. Now, from a workflow standpoint, the stain is not stable at 37 degrees for 24 or 48 hours. And so what you can do is you just take the plate out of your Puma system, add in your stain, and then go back in the Puma system to do your stain incubation, do your wash, etc. So the ability to remove this plate and put it back in really facilitates these type of assay steps. So here shows some representative images of tumoroids that were exposed to romidepsin and trametinib, and showing the decrease in cell viability. And this is a viability stain cocktail mix, calcium AM, thydenium homodimer, and Hirsch. The images were taken with the image-stress microconfocal system, in this case a 10x objective, and then we did the analysis for live cells and dead cells using the thydenium homodimer and calcium AM ratios. And then that allows you to look at the response to drug treatments. Interestingly, the paclitaxel EC50 was much higher than you normally see for the cancer cell lines, factor 100 to 1,000 higher. And that was very consistent with this patient's tumor response. Again, they were resistant to Taxol therapy, so we can recapitulate that disease resistance or tumor resistance to Taxol in these 3D tumoroid models. So next thing we did with these tumoroids is a metabolite analysis. The metabolism and working with Promega, they have a series of luminescence-based kits for looking at glucose, lactate, glutamine, glutamate. And the workflow is actually suits this type of assays really well, because from our Flowchips, you can do time point sampling. So in this case, we took 5 different time points, and from each of those time points, you have 32 organoids, 5 time points. The sensitivity of these assays is such that you can look at all 4. You can look at actually 5 or 6 different analytes from that supernatant if you want to, using these luminescence assays. And so in this case, we took the tumoroids, we incubated them with different compounds, and then selected samples at 4-hour time points. Another question that comes up, okay, well, you've loaded these tumoroids in there, how do you know how many tumoroids are in there, how many cells are in there? And so we're working with, again, Promega, they have a real-time glow cell viability assay. In this case, you have a cell viability pro-substrate that gets into a viable cell, and then it gets reduced into a substrate which is secreted, and then the nanoluck produces light, which is proportional to the number of live cells and the activity of these live cells. With the dead cells, you don't get any reduction of this pro-substrate to substrate. Live cells, the pro-substrate to substrate. And you can see here that when you look at spheroids, and this is looking at 4 different spheroids going from 1250 up to 10,000 cells per spheroid, you get a nice linear response of the signal here. And so this allows you to calibrate the number of cells, or the number of spheroids, if you will, you have in your well at the beginning of your assay. Okay, so we talked about doing the time course sampling. This, again, done automatically. You can select the number of time points you want. In this case, we chose 4, 8, 12, and 16 hours. If you're doing a faster dynamics, you can do every 30 minutes if you want. If you're doing slower, you can extend those hours. So it's a very flexible system. And then this is the results. These, again, preliminary results, but very intriguing. And what we're looking at here is the response of these tumoroids to paclitaxel and romidepsin treatment. So different concentrations here. And then looking at the time course of that. And so you're looking at the response of the metabolism response of those tumoroids at 8 hours and at 16 hours. And what you can see here is that the paclitaxel is increasing metabolism of the tumoroids at short hours for these higher concentrations. And we thought, well, that's very interesting, and it's actually been seen before with Taxol-resistant breast cancer cell lines. That adding Taxol to these before the Taxol actually causes cell death, you see this increase in lactate production, so increase in glycolysis of the cells. And then if you say, well, are you killing the cells? Or are you affecting them? You can look at viability of these cells, and this is after the 16-hour incubation. You can see there is an effect on viability, but there still is sort of a core of these cells which are still alive. So you haven't killed them all, but you definitely increase metabolism as a resisting the Taxol therapy, if you will. And then the last is immunofluorescence staining. In this case, again, using the PUMA system to transfer in this case, fluorescently labeled primary antibodies for ECAT-HERE and CD44, and then doing a wash step of those, and then imaging them. This is 20x water immersion lens and an image-stress microconfocal. And you can see really nice, clean, crisp images of these tumoroids. And then you can look at the concentration response of these. This is looking at 3 compounds that we looked at before in viability, romidepsin, trametinib, paclitaxel. Again, seeing the increase in the EC50 for paclitaxel, very consistent with viability. In this case, the viability we measured by the CD44 positive cells. You can see that the ECAT-HERE goes away almost immediately. So we have some more work we've been doing looking at trying to understand the EC50 of the ECAT-HERE response, and hope Marguerite and Matt's group publish some of that results in the near future. Okay, well, thank you for your attention. I think hopefully went through and showed the value of our PUMA system for doing automation with different organoid 3D cell model types, because it's an open platform, allows you to do a wide variety of type of assay protocol steps that we normally have to do manually. And with this unique Flowchips, our proprietary Flowchips, you can protect your 3D cell models during those steps. And then by using the NanoShuttle, you can then really improve those assays, improve your results by positioning them, doing more automated transfer of the organoids, and centering them, so it allows you to get that better imaging. And then with that, you can now think about doing this multifunctional profiling, where you're not getting a single data point from your organoids, but you're getting multiple types of responses from viability, metabolism, the dynamics of them, looking at biomarkers, et cetera, and really helping illuminate biological responses of your amazing organoid samples that you've taken so much time together. With that, I'd like to thank you and take any questions that might have come up.
Thanks, Evan. Could you tell us a little bit more about the Flowchip dimensions?
Yes. So Flowchips themselves are on a 384 well spacing. So the holder itself here conforms to SLAS ANSI standard. The wells are 3 millimeter diameter at the top, again conforming to the standard, and then the height is about 6 millimeters, which gives you a volume of 20 microliters in the well. And then at the bottom, this bottom chamber is 1.2 millimeter clear aperture, volume of 1 microliter in this bottom chamber.
Another question that we have from Gwen is, what is the maximum size of the organoids compatible with the PUMA system?
100 micron to roughly 1 millimeter. 1 millimeter organoid will fit in here. And then in terms of smaller size, we've done 50 micron organoids, et cetera. So you can go down to smaller ones. People often ask, can you do single cells, et cetera? Short answer is you can put single cells in here. We're actually looking at cell organoid interactions, et cetera, T-cell therapy, things like that. But the microfluidic channels are on the order of 50 to 100 microns. So your single cells will go down the channels. You might lose some single cells. You can put single cells in here. We have done. From an organoid sample, I'd say 100 microns to 1 millimeter.
One more question from Angeline. Have you tested the NanoShuttle with iPSC-derived organoids?
Yes, we have. We have different aspects. We have attached them to the iPSC liver cardiomyocytes. We have a paper that just came out from AstraZeneca where they co-culture iPSC cardiomyocytes with fibroblasts. And they were actually able to put the cardiomyocyte on top of the electrode magnetically and actually measure electrophysiology. So we have done also hepatocytes. We have actually, I think, a PubHub. We have a publication, a PubHub with Promega, showing that too.
So one question, this is for Evan. The 1.2 millimeter diameter for the space where the tumoroid or organoid sits, is it the complete well of the size of the well?
Again, the 1.2 millimeter is the bottom diameter of our protected chamber. The sample well is 3 millimeters at the top. There's slight change here, so maybe 2.5 millimeters at the bottom. But the clear aperture is, again, it's an injection molded COC plastic. So if you're doing any work with COC, 384 well COC plates are very similar to that. Thin bottom, 140 micron, thin COC bottom layer, so compatible with water immersion, oil immersion, any high content imaging you want to do.
So one of the questions is, have you used this with a bioprinter? So the question is like, how do you use a bioprinter with this system? So for example, with say with the NanoShuttle, is this something that you have explored, Glauco?
Again, there's different ways of bioprinting, but I'm thinking if anybody has used magnetized cells to bioprint, we have used. We have not. Of course, there's the magnetic bioprinting, which you basically use magnets to pattern the cells. But this could be, there's no reason why it's not compatible, depending on how you use. But a lot of the times, especially the extrusion type of bioprinters, they use spheroids. So you could magnetize the spheroids and you could dispense them through a nozzle without a problem. So depending on what your application should be, and also you can also mix and match, use the extrusion with the magnetic field to position better, guide the cells too. There's a lot of ideas that you can do using the two approaches.
Got it. So there is no time limit on like how long the NanoShuttle has to interact with the cells, because if you were to try to extrude it, right?
No, because usually if you're extruding, especially if you're extruding spheroids, often they make small spheroids and they extrude the spheroids. The nanoparticles is stuck on the actual cellular matrix of the spheroids. The spheroids stay magnetized for a long time.
Now, any of the organoids and that Glauco has been showing where you're using the NanoShuttle to form those, you can easily pick those up, the mag pen type of thing, and put them into our chamber to run your other assays. And then I sort of alluded to creating devices that you can actually do the formation. If you're interested in that, please talk to us. We are looking at that. Seems it's a natural next step for us. So love to talk to you further about what you'd be interested in doing.
This question is for Glauco. This from Matilde. Are the nanoparticles compatible with with formaldehyde fixation?
Yeah, it's usually they are. We use standard protocol paraformaldehyde or 4% paraformaldehyde to fix the cells. We do that quite often.
The question from Kune was, in the case of spheroids that are going to be treated with drugs, would the nanoparticles interfere in any way?
No, I didn't show this data, but we did quite a bit of studies looking at if there was any drug interference with drug interaction with cells, and there's none. We did a big screen with scripts and with cancer cells, and there's no difference.
Thank you so much, Evan and Glauco, and thank you, Jackie, for taking the questions in the chat. Thank you so much.
Thank you, Russ.
Thank you very
Russ.
much.
Glauco.
Thanks.